Okay, all right. So today it's my great pleasure to be here to share and then discuss our recent work by using a statistical approach and the computation methods to understand adaptive immune rupture receptor rupture. And so the human immune system plays significant role for protecting us from disease and infections. However, the immune system is extremely complex, especially for adaptive immune system, which responds and recognize the pathogen. Very simple is like, now it's allergy season, right? A lot of people have reaction to the pollen, and maybe you just get a cold a few days ago, right? So a lot of symptoms, those are due to your adaptive immune system react to those pathogens that including virus, like pollen and maybe cancer. So the adaptive immune system relies heavily on the receptor on the T cells and the B cells, which we call TCR and BCR. So standing for T cell receptor and the B cell receptor. So they both form extremely diverse ruptures. B cells are more closely related to antibodies. Antibodies were discovered in late 19th century through the more serum immunity studies. So the BCR actually they generated a B cell memory with the long-term memory about antigen recognition. TCR was identified in the late 1980s, so it was actually about a century later than BCR. But this also opened the door about the T cell-based immunotherapy that including TCR engineering therapy, and more importantly, help us to understand how T cell, how your adaptive immune system respond to the drug, to the disease. So for both TCR and the BCR receptor, structurally, they have three regions, which we call CDR1, CDR2, and CDR3 regions. CDR stands for complementary determining region. And among the three regions, CDR3 region is the most diverse region that generated based on the VDJ recombination through the T cell and the B cell development. Therefore, most of the time when we talking about the TCR and BCR sequence, we mainly talking about the CDR3 region sequences. Okay. Therefore, for the bulk TCR or BCR sequences, like most of the next generation sequencing, we have the primers. So we put the primers on the VDJ gene junctions. And then we will have the FASTQ data. After the pre-process, normally we'll have the sequences for within each of the rupture sample. However, the adaptive immune receptor rupture is super variable and high dimensional. It usually generates thousands to millions of the sequences within even one sample. Okay, so this actually brings us the challenge about data analysis, right? Because it generates high dimensional data and highly variable due to the adaptive immune system. Because even for the healthy donor, within 2 days, if we get blood sample and then we sequence them, you can see that the sequences actually change a whole lot. Normally, the overlap sequences is as low as less than 10%. So the traditional data analysis, like differential testing, want to be able to apply it here. Okay. In addition, most of those sequences actually has unknown antigen specific information. Therefore, in the last 1 decade, people have been dedicated doing figure out the best way to do the data analysis. This one is a diagram of our group have been done so far for the last 10 years. So we started doing the data analysis by, providing to figure out what is the global matrix to describe the rupture. So for example, we develop like a chronality, T-cell convergency to describe the diversity of the rupture. However, the global matrix is something like, you use mean and standard deviation to describe, like, for example, in the classroom, we have, students and the faculties here, we want to describe the age, right? If you use mean and standard deviation, certainly you can use that. But, as statistician, that also has a limitation because we lose the granular level information of the repertoire, right? So later on, we also consider about the VJ gene usage. So we applying the similar method as traditional feature selection and classification to understand which VJ genes actually more highly expressed or used in cancer patients versus non-cancer patients. But immune receptor evolve and then diversify. That's why we think about, is there any way we can think about the more granular level? Especially people already found out that for the TCR sequences, if they have high similarity, they intend to recognize and binding to the same antigen. And for the BCR, even for small somatic mutation, we can see there is a change in the sequence. Therefore, we think about, can we build a network based on the sequence similarities that bring the first project I'm going to talking about is the network analysis of immune repertoire, which we call NAIR. This was motivated by a project, it's about superlucil T. We want to study if the superlucil T will induce the long-term B cell memory. Superlucil T is a FDA and EMA approved therapy for prostate cancer patients. So briefly, superlucil T it's a kind of vaccination type of cancer immunotherapy. Basically, each patient get their blood sample draw, and then it cultured. 2 days later, the final product is generated, and then this final product is injected back to the same patient. And then 2 weeks later, the patient get another blood draw, and then the blood sample get cultured. And typically, the superlucil T is 3 injections. That's the whole process of treatment. So for this particular study, what we were interested in is like I said, we want to check the B cell memory. So we have 2 cohorts of the patients, prostate cancer patients. The first cohort was the patients, they never received superlucil T, so which we call naive patients. The second group of patients is retreated group, including the retreated group of patients. So those patients were actually treated about 10 years ago, but their disease got progressed. So then they got retreated. So we want to compare those 2 groups of patients. We first use the chronality, which is the way to assess the diversity. We already found that the patients who were retreated, you can see that even at the baseline, you can see that the chronality is much higher. So the baseline is before the treatment, they received the treatment. So when we say chronality is higher, means that the patient's repertoire is getting more focused. So then we were very interested in what really happened. Like I said, a global matrix is important. It's like a biomarker, but then we really want to look into that, right? So then we performed the network analysis. We think initially we were thinking about to just correlate, cluster the sequences which have high similarity, and then eventually we build a network analysis. But what does network analysis mean in the repertoire? So we can just mimic this with as a social media like a Facebook or Instagram. Okay, so basically here the node stands for the BCR clone, and distance is the number of letters difference between the 2 sequences. Okay, the age is defined as only 1 change between the 2 sequences. Of course, you can consider about 2 differences or no difference. So this is a little bit arbitrary. And distance matrix is the distance matrix, pairwise distance across the among the clones. The attributes basically is the metadata. The metadata could be, different types of specimens, such as if it is coming from blood sample or tumor sample, if the sample is coming from baseline or it is coming from the second post-treatment time point. So it really depends on the needs. And the cluster is a group of the clones which have indirect or direct connections. Okay, so to build a network, 2 major components. The first one is the distance matrix. The second one is clustering method. So we don't really spend too much time on the clustering method. We just use existing methods in the R package, and then we did some sensitivity analysis, and turns out that they all perform very similar. But distance matrix here matters. We consider two different types of distance matrix. The first one is called Hamming distance. Hamming distance is easy to compute. But it has its own limitation because it can only kind of explain the transformation from the biological sense. And also it can only deal with when the sequences are in the same length. So if the sequences are not in the same length, we have just padding in the end to do the calculation. So you already see the limitation for the Hamming distance. For Levenston distance, it's more interesting because it can count for different biological transformations, so that including transformation, insertion, and deletion. It can deal with, of course, it can deal with the sequences with different lengths. But because it's flexibility, then it also create challenge for calculating, the high dimensional distance matrix. It takes us like for the very first project, it took us actually about 2 months to calculate 100 samples just because the large distance matrix. But recently, one of my students developed method to improve this performance. But due to the time limit, I'm not going to illustrate those details. If you are interested, you can look up online. So by applying the network analysis on the supercell T project, we actually found it very interesting. So the first one, sorry, those two network were based on the samples from retreated patients and naive patient. Okay, and this is just one patient each. And then this is the baseline. So you can see that before the treatment, okay, so the network is already very complex. Here we only plotted the clusters with at least 5 nodes. Okay, and then if it's more than 1,000 nodes, we only plot the top 1,000 nodes. Okay, so to summary, across the two cohorts, we found that the retreat patients always have large cluster and then more number of the clusters in the network. Okay, we also look them longitudinally. So this two network was looking at 6 different time points from baseline all the way to 1 year follow-up time. Okay, so you can see that. So the network is starting. Oops, sorry. The network is starting to. Getting more complex, but still comparing to the retreated patients is not as complex as. Okay, and then here's the summary statistics. Okay, so the takeaway based on this motivated example is that, okay, from biological point of view, we do see that the supercell T induces long-term T cell or B cell memory for years, right? Because remember, those retreat patients, they were first treated 10 years ago. Okay, and then more importantly, we found out that the network analysis probably can represent a novel method for us to study the adaptive immune receptor rupture. And we actually applied this network analysis on multiple projects, including lung cancer patients and a single cell data set. Okay. So then, because I'm working in the collaborative environment, and then after we have done a couple of projects, we start to think about, okay, instead of just build up functions, why don't we just build a software so people can use that widely? That's why we have our Neo software published in R. So this software is a very flexible software. It can deal with not only the box sequence, but also the single cell. So if you are familiar with the Tanks genome sequencing data, so it can also deal with the sequence TCR, BCR generated based on the Tanks genome single cell data set. It can be applied to both TCR and the BCR sequence, and then can deal with different types of distance and then the clustering methods. More importantly, we also develop 2 other pipelines, which we can identify the shared clones across different time points or across different samples. We also develop the software, the pipeline, which can identify the disease-associated clusters. Okay, so here's an example how we apply this network analysis on a group of COVID-19 patients. So this data set was the paper was published by a group of European researchers in late 2020. That was, during the pandemic year. So we were able to contact the author, got the data. So basically, this project involved three cohorts. Two cohorts are COVID-19 patients. One is recovered patients group, and then which they had a mild COVID-19 symptoms. They didn't actually receive any medical intervention. And then the other group of COVID-19 patients, they were very severe, and then most of them actually, unfortunately, died. And then there was also a cohort of healthy donor. Okay, so we're applying this our pipeline. This is just an example of finding disease-associated cluster workflow. Okay, so the nice thing about this workflow is that even though we call this is disease-associated cluster, but the idea behind this is like it can, COVID disease associated. It could be also cancer associated. Also can be identified as, say, respond associated. So it's very flexible. It depending on how we want to define the outcome. Okay. So by using the pipeline, we were able to identify the TCR clusters, which were like COVID only. So that means those TCR sequences, they only present in the COVID-19 patients. And also the COVID-associated TCR clusters. So the associated clusters, that means, okay, those TCR clusters, they present in both COVID-19 and healthy donors. However, it's more frequently presented in the COVID-19 that we call COVID-associated TCR clusters. So now, if you recall, I mentioned earlier, because of very high variable the feature of the TCR sequences, we cannot really perform differential testing like we usually do for gene expression data, right? So now, because we generated a global membership based on the TCR cluster, so we can perform differential testing based on the cluster level. So in the end, we were based on the COVID-19 or only or associated clusters, we perform downstream analysis. So in the end, we identify, over I think a total about 20 such TCR classes. They are associated with COVID-19. Okay, so they were statistical proved highly, abundance in the COVID-19 patients. Okay, but why are they useful? So that's probably the question people ask, right? Because as we're doing this, we want to make this more translational, right? So we want to approve what we found is really meaningful. What we did is this: we tried to validate them in a public available data set, which called MIRA. MIRA is a curative data set. It was a wet lab validated TCR antigen information. So all the antigens are represented for the SARS-CoV-2 epitopes. So basically, it's like you can think about the TCRs in that database all have been verified as the SARS-CoV-2 epitope specifically. Okay, so we found, for example, the first, I'm sorry, I apologize, the small font, but the first cluster, for example, we have over 800 TCRs in that cluster. Among those 800 clusters, more than 80% of those TCRs will actually exactly match with the MIRA database. That means this TCR cluster, we can claim that this TCR cluster is SARS-CoV-2 epitope specific TCRs. Okay. But very interestingly, there was few of those TCR clusters has no matching at all. So what does it mean? What happened? Do we think, so are those TCR clusters are useless? Then bring us to think about, can we predict if the TCRs are really predict to bind or recognize the antigens? Okay, so that's the motivation for our second project is we want to build a prediction model, excuse me, to think about the prediction model to build the TCR peptide interaction. Okay, so we can consider the network analysis, which is more sequence similarity, is more unsupervised way to identify if there's a TCRs recognized antigens. And now we want to build a supervised method to learn a prediction model. Okay, so this is our overall pipeline. So basically, you can think about it in our as many other prediction model. You have your inputs, you have your feature, right? You want to predict your outcome. So here the prediction is more classification, because our outcome is more like, you have tons of, like I say, 5, 10 of those antigen peptides that you are interested in. Okay, then you want to predict which T cells binding or recognize this antigen peptide sequence. Okay, so the input data basically the TCR sequence along with their VJ gene information and then HLA type. Okay, well, why HLA type is important here? So HLA type, HLA stands for human leukocyte antigen. Okay, so TCR cannot recognize antigen if it was not presented by specific HLA molecules. So that's why HLA is very important in the prediction model. However, the challenge is that in our current data, in our current situation, a lot of time we do not have the HLA information for the individuals, or sometimes even we have limited information. So later on, I will show you how our model robust if even without the HLA information. Okay, so basically here, when we build this prediction model, 2 major steps. The first step is feature engineering. That's actually the main part of our prediction model. Second part is prediction, which is related for straightforward. Okay, so now feature engineering, why it's important, especially the sequence embedding module. The reason is that for both peptide sequence and then TCR sequence, you can see that they are all letter-based amino acid sequence, right? So they are, the HTTD, whatever, those kind of sequence. People already have built a prediction model just based on sequence. But if you just based on the sequence, letter-based sequence, you basically treat them as a categorical variable, right? So you just think about it, it's very difficult to deal with. That will actually lose the, kind of decrease the performance of your prediction model. So what we want to do here, the first thing is we want to embed those letter-based sequence into numerical value. Okay, so here we introduced Ashley factor vectorization. We are not the first person to use that, but we use it more smartly. Okay, so Ashley factor represent over 500 different amino acid properties in 5 properties here, which you can see on the top, 5 of those properties. So we also add an average of those properties. So in that way, you can see for each amino acid, actually you can decode this into a vector of 6. Okay, so now think about if you have a sequence of like A, G, B, K, blah, blah, like that, right? So each of them you can decode into a vector. Now you just put them together. Then you have a long, very long vector with a numerical value. Okay. When we have different sequences, what we do is we adding the 0 padding in the end to make sure all of them are in the same length. Okay. Well, of course, by doing this is good, but then it's also limited. First limitation is you can see that the same amino acid can present in different locations, right? So if you treat them the same, of course, we lose the position information. Therefore, we introduce the positional encoding here, just adding a small amount to try to distinguish their different location on the sequence. Okay. Well, after we have this embedding, the positional encoder output, we can already treat this as input for our prediction model. Okay, but we recognize that by adding the 0 padding in the end and also adding the positional encoder, this also introduce like a high dimension and also noise. So we consider use the autoencoder to standardize this numerical vectors and also to do the dimensional reduction to remove the potential sequence noise. Okay, again, so we can stop right here. We can just use the positional encoder after autoencoder as input. But then we realize that, okay, in the public available data, we already know millions of those TCRs have no antigens. Why why don't we use those information? Therefore, we bring in encoder decoder. The encoder decoder, you can just think about it's a sequence to sequence learning model. Okay, so basically in the public available data set, we already know the structure between TCR and the peptide sequences, right? So now we bring in this information and we by using the encoder decoder. So, but try to avoid the data leakage. Later on, we try to use also complete external validation to make sure about there was no data leakage. Okay, so the last part is we want to bring in our network analysis here. So remember, we build a network based on the similarity, right? So now think about, you can build a network based on the TCR sequences, right? Now, for each node, you can add in the information by adding the encoder decoder information on the node. Okay, then by using graph neural network, we can build another layer of the TCR node embedding. Okay, so by doing this, we try to capture both the topological structure of the network along with those numerical embedding TCR sequencing information. Okay. For the categorical variable that including HLA and VJG, it's very simple. We just, I wouldn't say very simple, but straightforward. We use one hot encoder and the auto encoder. Okay. Now goes to the prediction part. So for the prediction, actually we tried a different way to do prediction, including the traditional, say, random forest, traditional machine learning method, and also some other deep learning method. But in the end, we decided to use Bayesian fully connected feed forward. The reason why we consider about the Bayesian method here is that for TCR antigen recognition, that whole process, it's actually not one versus one. What does it mean? So we know that multiple TCR sequences, they can recognize the same antigen. But from the other point of view, one TCR sometimes can also recognize multiple different antigens. So, but when we build a model, we actually consider about the 1-to-1, right? So by using the Bayesian method, we want to introduce the uncertainty for this whole prediction method. Okay, so in the end, what we have is for each TCR and then peptide, you have a posterior distribution, right? And for each posterior distribution, we draw 200 samples. And then the median probability, we use the median probability as a decision. So we choose the highest median probability as our decision. Okay. All right, so we use public available data set to train our model. Those are the 4 databases, largest database right now in the world about TCR antigen recognition. We use IEDB, VDJDB, and the MarkPath as our training model and training data sets for and use the mirror as our external validation. And more importantly, we use those 3 data sets as the training for the embedding, so that when people want to just get the TCR embedding, numerical embedding, they can just use our trained model. They don't have to retrain the model. So even if their goal is not to do TCR antigen prediction, if they just want to encode their TCR sequence, they can just use our sequence embedding module directly. Okay, so for the public available data sets, right now, unfortunately, most of those antigens still are viral antigens, so that including like HIV, herpes virus, and also SARS virus too. But the database is growing right now. We also start to see more and more tumor antigens because I'm mainly working in the cancer research. That's why for us, tumor antigens are very important. Okay, so now the exciting part is coming. So in our in-distribution, this is the prediction model, prediction performance. I'm sorry, the top one's covered. But this is basically the prediction performance of our in-distribution data. So no doubt, when we have full input, which is the TCR and with the HA information, of course, performance the best, which is the rightmost 3 columns. Okay, but very interestingly is that even if we only have the TCR sequences only, you can see that the one I circle and this one, you can see their performance is always already comparable with this input, which you have the TCR plus HA. Remember, I mentioned HA is very important in the TCR antigen prediction. Okay. However, due to the many reasons, a lot of time the HA information is not available. So, but you can see based on our model, we can see that with our embedding model, we can have very comparable performance of TCR only prediction. Okay. We also want to check the robustness of our model because it's more classification kind of methods, right? So, when you have 5 classes versus you have 20 classes, of course, the model performance will be different. What we found here is that even we increase the, classes from 5 to 20, even the performance you can see in general decreased a little bit, but our full model always performed the best and then very robust across the number of the classes. And we also want to check in the external data, which is the mirror data itself. It's very imbalanced data. And then, so just quickly, this is AUC of each of the peptides that we want to predict. You can see the AUC of ours. I think here we use the, Here we use the PR AUC, so it's a precision recall AUC. Instead of we using the traditional AUC of ROC just because it's imbalanced. Anyway, so you can see the performance is pretty good here, right? And then, well, you probably noticed the confusion matrix. This one is a little bit off. The reason is because this is not one peptide, it's 2 peptides. So it's a classes. That's the reason introduce more noises. Okay. We also compare with other existing methods. So if you develop any bioinformatics tool and write a paper, you know that there will always be you will be always asked to provide a comparison with the benchmarks. But anyway, so we comparing existing methods. So there, in general, our performance is much better than theirs. So remember that. Okay, so our predictions based on the Bayesian, BFNN, right? So it's a Bayesian method. So to introduce the uncertainty here, we use the Valin plot to plot the posterior distribution. And then the top one shows the true label, and the bottom one shows stand for the false label. When I say true label means it's you really want to predict your TCR binding to those particular peptides, right? And then the last column is our method. You can see the Valin plot always stick on the top, and then and for the bottom one, we always expect this is always stick to the bottom, close to 0. Okay. All right, now let's going back to this one. So you recall we have this table by using the network analysis, right? I mentioned that, okay, here are some of those clusters showing very low, matching with the mirror data set, right? So now we want to say, can we do the prediction of the TCRs within each of those clusters? So we apply our, PEP TCR net method to this data set. Okay, so of course we have to train the model, and then this is confusion matrix, and this is the AUC curve. Okay, so now what I want to show here is just look at this one. So this is the clusters which corresponding to the previous slide. Okay, so you see cluster 3, 1, 3, 9, and so on, right? Okay, so the color code stand for, if this is blue, means they are exactly matched with the outcome, right? They are exactly matched with the database. And then the green means they were matched and we cannot predict them. And then the orange one means that, okay, they were not matched, but we are able to predict them, recognize the antigens. Okay, it's very interesting. So the 3 of them I highlight here, remember this was 0 and this was 0% matching. Now we were able to, identify some of those TCRs in the small clusters. They were actually having the SAS virus 2 epitope specificity. Okay, so to summary, by using the PEP TCR net method, we actually can, perform better performance in terms of prediction, and then we can actually nicely we incorporate the network similarity into our embedding step, and then scientifically, we can identify those previously unannotated TCR antigen interactions, right, as shown in the previous slide. Okay, we still have some time, so I want to share with you guys. This is the ongoing project, so this is unpublished, but it's very exciting. That's why I want to quickly share with you guys. This is actually, again, it's still about the TCR antigen prediction, but we actually want to integrate the TCR and the gene expression. Because those data I'm going to focus on the thanks genome data set. So I think it's auto battery. Sorry, this is auto battery. I hope I can raise up my voice. Uh, uh, you guys can hear me. I think it's good. Oh, okay. Oh, great. Thank you. Yeah, it's all. Okay, great. Thank you. Oh, fast. That was fast. Okay. Yeah, so this is talking about the thanks genome data set. So thanks genome data set, let me just move on to the next slide. I want to give you some overview about what thanks genome data set looks like. So thanks genome data set is so this is single cell. So now it's at each level, it's actually a cell level. And then each cell has both TCR sequence and also gene expression data. So this is more about the integration of multi-modality data, right? Because they are completely different. Okay. I'm not going to go through the details, but I want to show you here is that the major part here is we want to integrate the gene expression and then the TCR sequence, okay, which we propose two different ideas. One is actually using the GNN, graph neural network, which we still think about building the network based on TCR sequence. Okay, then on the node, we actually incorporate the gene expression data. Then we can generate the embedding information. Okay, the next one is using the encoder decoder model. So now we just can see about the two inputs. One is the gene expression data. The other is the TCR sequence data. Okay, so here's the UMAP of those the results. Okay, so if you're familiar with the thanks genome data set, UMAP is a very traditional way to present the data to see their clusters. Okay, so basically the top row are the, doing the UMAP, and then the color code is clustering by just the latent clustering, unsupervised clustering. The bottom one, those figures, those were color coded based on antigen peptides information. Okay, and the first one is just we consider gene expression data. Second one is we consider ED integration, and this is a GN integration. Okay, so from visually, you already can see that by using the integration method, this cluster should very correlated with the antigen information. And then we also show this in Harvard, our collaborator from Harvard have the mouse data. And then for this mouse data, actually, we have no antigen informations. So similarly, our way to organize the figure, but you can see basically, I want to show you here, if we're only using the gene expression data, it's very difficult to distinguish between these two known antigen information, right? But after we do the integration, no matter we use ED model or we use the GNN model, you can see the cells actually separately well by different antigen information. Okay, and then here is the numerical value to compare across different donors and information. Basically, this matrix, you can just think about the larger number shows more consistency. Okay, this is an ongoing project. We're still working on the last part about our, how we can improve the antigen recognition modeling and then working on the prediction part. Okay, so final note. So the takeaway is that, so today I actually discussed about the roadmap of our groups, how we're doing this, how we identify the antigen, TCR antigen recognition by computational method from the unsupervised clustering by the network analysis. and then to by build the prediction model. And then in the last part is integrate the T cell and antigen, T cell and the gene expression data. That is more interesting biologically because we can also think about the cellular transcriptome states information. Yeah, that's it for my talk. But last but not least, I really want to appreciate, show my acknowledgement to my long-term mentor, Dr. Feng, who now moved to Fred Hatch Cancer Center, and also my collaborators, and also my lab person, postdocs who work very hard on those computational approaches, and also the funding agency from NIH, NCI, and then also NLM, and also the uses of internal funding opportunities. Yeah, thank you.
Okay, so can someone have a layman's question? It's really impactful work. My question is, in the second project, the supervised prediction part, how do you feel about the accuracy of the training data? Because when I try to explore some supervised like training or prediction tasks, sometimes I find the training data cannot be very high quality, and this can be not very ideal. And also, are there any like quality control preprocessing steps you would recommend?
That's good questions. So, okay. So first of all, for our projects, tuning actually that takes us a lot of time. But what we found out is that the more robust in the training will help us to have for better performance in the validation testing. And then I didn't present very clearly. Actually, in our analysis, we have the data split. So remember the database. Let me see if I can going back to the, 1 second. Let me just going back to the slide here. You actually hit a really important point here. So when we work on this project, so it took us like 2 years to work on this project. Actually, the data, how to deal with the data took us forever. And then like the data currently we're working on the third project. Actually, the most challenging part is about how we split the data. And so that's why you can see that we try to generate as robust as possible data set. And then second of all, I have to say that that's my personal experience. Deep learning model in terms of prediction doesn't really help a lot comparing to the traditional machine learning. I know maybe a lot of folks here are working on the deep learning part, but that's my experience. But I figure the most important here in our case is the deep learning did play a significant role when we're doing the feature engineering step, the embedding part. So I figure it's like, when sometimes in statistician, we say that garbage in, garbage out. So when you actually have your embedding, it didn't have really good, nice embedding, the features. I mean, you don't necessarily have embedding because if you are not working on the feature, like a sequence data. But in general, I would say if your features, the feature engineering step, you didn't do well, then your most likely your prediction part will fail in the validation step, right? And then the other thing I want to add here is actually when we have the data, we split into, if I remember correctly, is we split into two parts first. So the first part, then we split into training, validation, and testing. So basically, each of the large data set we split into three. And I think this, sorry, I didn't really write the code myself, but I think we had a lot of discussion. In the end, our split was like 6-2-2. So 6 training. So you can think about it. So 60% is for training and 20% is validation and the 20% for testing. This is actually only happened in only this part, okay? And then we validate again externally. So those steps are very important. That's why make our performance really good, just because we try to make our model as robust as possible. Because as you know that, especially for deep learning, right? The parameter tuning, right? You can make it perfect, maybe just in your training, but then your validation could be really bad. So you want to balance out. Yeah, that's my suggestion. Thank you for the question.
Simple question for the COVID-19 patient. You said the patient, they are observational patients, right? They did not have any existing condition or only COVID-19 one.
Oh, you mean the COVID-19 patients? Yeah, those, yeah, that's actually you brought up. So, traditionally for me working in the cancer center, we actually, let me see if I can find the cohort information here. So I work in the cancer research, as I just mentioned. So a lot of time we actually have very nice sequence data, very nice study design, because it's all coming from clinical trial, right? So there's a lot of, we can get rid of a lot of confounding factors, right? But for this cohort, it's steep, especially it was in the early, age of the COVID, right? So those were just really they randomly, kind of recruited from the hospital or from the like so they so for some of those patients we have longitudinal samples. Some we only have one sample. Okay, but we actually distinguish them. So our analysis was different from the original paper was that we don't really just say distinguish them from like a mild COVID-19 patients versus severe 19 patients. We didn't do this way. Okay, so what we distinguished was based on their symptom. So we kind of distinguish them between like if the sample was collected when the patients, well, I wouldn't say patients, the individuals have active COVID symptom versus they already recovered. Yeah. So that way, you just think about it because your immune system react differently when you have the symptom versus when you already recovered, right? So we basically, that's why when we do the analysis, sorry, I didn't really say that very clearly. If you see here, we actually compare its active samples versus healthy donor, recovered samples versus healthy donor, and recovered versus active samples. So even for the even for the COVID patients, right? Recovered sample versus active sample, you can see that we can still identify the TCR clusters. Their abundance differentially different, because that means like your adaptive system is react to the virus. Yeah, it's a very complex system. Because even for the healthy donor, we collect the samples even 1 week apart. You found that their sequences change dramatically. They only have less than 10% of sequences, stay for within 1 week. That's for the healthy donors, because you don't really know what happened in your body. You look maybe healthy, but maybe your body just experience a fighting with some allergy. It could happen like, okay, your family member got cold, and you say, oh, it's amazing, I didn't get the cold, right? You thought you were not infected. It's not necessary. You might all get infected, but you didn't notice that just because you do not have the symptom. That's why we call a symptom. Remember during the COVID time, right? Some people go to get tested, right? They say, oh, I have COVID, but I don't have any symptom. Well, this is just like everyone present differently. Yeah.
Any other question? Well, quick one about your like the graph structure. So the graph you mentioned is the GRN, the gene regulatory network. Is that the one?
Which you mean the graph neural network?
No, there's the graph structure. Before you move to the next slide on the Bayesian neural network.
Oh, okay. Let me see.
First by the gene, like the cell similarity or what's called genericity.
This one?
Yes.
Oh, okay. Yeah. So this one doesn't have any gene expression data only yet, because this one is just purely based on the TCR sequence. So what we do here is you can consider we build a network based on the TCR sequence, right? As I mentioned, the nail package, right? So each node stand for the TCR clone, or so you can say TCR sequence, right? And then what happened is that we now, for each of those nodes, we incorporate the information is from the embedding from this encoder decoder. And then we put this, so if you see this is a little vector, so that numerical vector is coming from the embedded, the latent space. So this is coming from the encoder decoder output. So basically we use the graph neural network basically is combining the network structure with the encoder decoder embedding. So just like adding another layer on top of it.
Okay,
Yeah.
you.
Thank you.
Any other